So, in this session we are going to cover the culture media and the culture methods. So, as we know that microorganisms require energy for their survival, the microorganisms require energy for their growth, they require energy for their replications. So, this energy can be obtained from different nutritional supplements. So, they require nutritional supplement, they require nutrients, nutritional requirements for their growth, for their survivals. So, the nutrients that are required for the growth of microorganisms is called as culture media. Media required for the growth of microorganisms, media which allow the growth of microorganisms or media which favors the growth of microorganisms is called as culture media. So, in this lecture we are going to study different types of culture media and different types of culture methods. Okay. So, when we study the microbiology of any sample or when we study the microorganism of any sample, there are certain things or there are certain steps that we need to follow. So, first step is inoculation. Inoculation, it is a process of transferring the microorganism from one place to another place. It is the technique by which we can transfer the microorganism by using suitable technique into another medium. So, sample is placed on a sterile medium providing microbes with appropriate nutrient to sustain the growth. That means, we transfer the microorganism by using certain inoculation loop or nichrome wire loop in another medium for their growth. The selection of proper medium and sterility of all tools and media is important, because if the media is not sterile or if the tools which we are going to transfer the microorganism into another media, if it is not sterile, then the false interpretation of the result can be possible. The microorganisms which are present accidentally into the media or the microbial contamination on the inside the media or onto the tool may interfere into the result. That is why the sterility of all tools, whether we are using apparatus, glasswares or we are using media, that has to be sterile. So, first step in this process in the study of microbiology or study of microorganism is inoculation, where we transfer the microorganism by using certain technique into another medium. Then the second step is incubation. After transferring the microorganism has to be grown into the media or onto the media. So, incubator can be used to adjust the proper growth condition of a sample. Already we studied that there are certain environmental factors that are required for the growth of microorganism. So, incubator, it is an instrument which provides the favorable condition for the growth of microorganism, like temperature. So, there is need to adjust for optimum temperature and gas content for the growth of microorganism. So, incubation produces a culture which is visible, which is having visible growth of the microorganisms or on into the medium. So, during incubation, microorganisms grow inside the media or onto the media. After incubation, there is a next step, that is isolation. The end result of incubation and incubation is isolation. The isolated colonies or the microbial growth can be observed. On the solid media, we can observe the growth of microorganism in the form of colonies, means we can observe the colonies onto the media. Like here, this is the inoculation of microorganisms onto the solid media. So, in the Petri plate we can see the colonies can be observed or the microorganisms can be observed in the form of colonies, where in this case if the microorganisms are inoculated in the liquid media, then we observe the growth of microorganisms in the form of turbidity. So, on solid media we observe the microbial growth in the form of colony, in liquid media we observe the growth in the form of turbidity. If crowded colonies are observed, then subculturing for further isolation may be required. If we found that the growth of microorganisms there is too much growth of microorganisms, or if we cannot distinguish between two different colonies or colony characteristics, then subculturing is required for further isolation. After isolation, there is a next step, that is inspection. Once the colonies are isolated, that colony can be inspected either macroscopically or microscopically. The macroscopic observations might be the note of color of the colony, texture of the colony, size of the colony, etc. And microscopic observation can be done by using different staining technique or the cell size and different motility parameters, whether the microorganism is a rod shape, whether it is spherical shape, whether it is motile or non-motile, that can be done. After that, the identification of microorganisms can be done by using different biochemical tests to differentiate between microorganisms from similar species. For example, we can use McConkey's media in order to differentiate between gram negative bacteria and gram positive bacteria, and apart from that, the gram negative the McConkey's media is selective for gram negative only. So, microorganisms which can grow in McConkey's media, they are gram negative, but it can be also used to differentiate between lactose fermenting and lactose non fermenting microorganisms. So, that can be done by using different techniques. So, in order to study the microorganisms or in order to identify the microorganisms, they must be grown on particular media. So, by appropriate procedures, they have to be grown separately or in isolated forms on the culture media or obtained as a pure culture for study. So, original media can be, it was used by Louis Pasteur from the urine or meat of the broth. So, liquid media produce diffuse growth and solid media produce discrete colonies. So, for the growth of microorganisms, certain media is required. As already discussed, the media required for the growth of microorganisms is called as culture media, or it is also called as nutrient media. So, nutrient media or culture media, it is a media required for the growth of microorganisms. When microbes are transferred into the culture medium or nutrient medium to initiate the growth, they are called as inoculum. Means the amount of microorganisms or amount of samples that is inoculated in that media, that is called as inoculum. The microbes that grow and multiply in or onto the culture media is referred as culture. So, those microorganisms which grow or which is transferred onto the media is called as culture. When we talk about culture, that means it is a suspension of microorganism. When we say mixed culture, it is a suspension of multiple microorganisms. When we say pure culture, it is a suspension of particular species of microorganisms. So, there are different requirements for the growth of microorganisms as discussed in previous lecture: moisture, pH, oxygen, sterile medium, temperature, all these are the requirement for the growth of microorganisms. We can use agar in the culture media, but agar does not have any nutritional value, it just used to solidify the media, because the agar it liquefies at 100 degrees Celsius and remain liquid until the temperature falls up to 40 degrees Celsius. Different petri plates, slants and stabs can be prepared by using agar in the culture media. So, colonies are the macroscopically visible collection of millions of bacteria originating from single bacterial cells. As discussed, the bacteria they divide by binary fission. So, after certain period of time, they divide in such a numbers which can be visible originated from the single bacterial cell. So, the culture media are classified into different classes based on their consistency. They classified as solid media, liquid media or semi solid media. Second classification is based on the constituents or the ingredients which are added. They are simple media, complex media, synthetic or defined media and special media. Again, the special medias are further classified as enriched media, enrichment media, selective media, indicator media, differential media, sugar media, transport media are the medias for different biochemical reactions. Apart from that, based on the oxygen requirement, the medias are classified as aerobic media and anaerobic media. So, if you see in detail, the solid media, they contains 2 to 3% of agar. As we already discussed, agar do not have any nutritional value, but they can use to solidify the agar media. The colony which are observed on the solid media are the microbial growth which is observed on the solid media is in the form of colony, and the colony morphology, pigmentation, hemolysis can be observed on the solid media. The example of solid media is nutrient agar, blood agar or McConkey's agar. These are some of the examples of solid media. The liquid media, they do not contain agar, hence they are liquid in nature, and for inoculation preparation liquid medias can be used. For blood culture, for isolation of pathogens from the mixtures, the liquid medias can be used. We can observe the growth of microorganisms in liquid media in the form of turbidity. So, in solid media we observe the growth in the form of colonies, and in liquid media the growth is observed in the form of turbidity. Example is nutrient broth, McConkey's broth, etc. The semi-solid media contains less proportion of agar. For example, 0.5% of agar can be used in semi-solid media, and the use of this media, it can be used as a medium for motility of the microorganism. The simple media or basal media, examples are nutrient broth or nutrient agar. The nutrient broth consists of peptone, meat extract or sodium chloride. To adjust the tonicity, sodium chloride can be used. If we add agar into it, then it becomes nutrient agar. So, basic difference between nutrient broth and nutrient agar is presence or absence of agar. The complex media, they are media other than basal media. They have some added ingredients like beef extract, meat extract or yeast extract. They are rich in energy, carbon, nitrogen and sulfur. They are provided in the form of proteins in order to enhance the growth of microorganisms. So, most of the microorganisms cannot utilize protein directly. So, partial digestion by acid or enzymes reduces the protein to shorter chain amino acids called as peptone, and this peptone can be utilized by microorganism for their growth. It provides the special nutrients like vitamins and organic growth factors. That means, the basic difference between basal media and complex media is addition of some added materials or additional nutrients. The synthetic media or defined media can be defined as the media prepared from pure chemical substance and its exact composition is known. That is why they are called as defined media. Their content or their composition is defined. For example, peptone water, it is 1% of peptone plus 0.5% of NaCl in water. Then enriched media like substances like blood, serum or eggs are added to basal media, and they are used to grow the bacteria that are exacting in their nutritional needs. For example, blood agar or chocolate agar, they contain some enriched nutrients in the media to facilitate the growth of microorganisms. So, these are the example of blood agar or chocolate agar. Enrichment media, these are the liquid media used to isolate the pathogen from the mixed culture. The media is incorporated with inhibitory substance to suppress the growth of unwanted microorganisms. Means these media are suitable or they are selective for particular type of microorganisms only. They do not allow the growth of unwanted microorganisms. For example, selenite F broth for isolation of Salmonella and Shigella, then alkaline peptone water for Vibrio cholerae. So, in this media, the other bacteria than Salmonella and Shigella will be suppressed. Then there is a selective media. It is a media which is selective for particular type of microorganism. Means this media has inhibitory substances which prevent the growth of other bacteria. That means it is selective for particular category of microorganisms only. For example, McConkey's media, it is selective for gram negative bacteria. It will not allow the growth of gram positive or other bacteria in this media. Then thiosulfate citrate bile salt sucrose media, TCBS, which is selective for Vibrio cholerae. Then Lonstein-Jensen media, which is selective for Mycobacterium tuberculosis. Wilson and Blair media, which is selective for Salmonella typhi. Potassium telluride media, which is selective for Diphtheria bacilli. And these are the typical colony morphology on TCBS agar media as follow. The Vibrio cholerae forms a large yellow colony, whereas this Vibrio parahemolyticus, it forms colony with blue to green color centers. Then Vibrio alginolyticus forms large yellow mucoidal colonies. Then Vibrio harvey fishery, they form grayish green to bluish green colonies. We show luminance in dark. The older colonies fail to show the bioluminescence. In protease, it produces partial inhibition if the growth of colonies are small and yellow to translucent. Pseudomonas, the partial inhibition, if the growth occurs, the colonies are blue. So, these are some selective medias which can be used to grow a selective microorganism in it. So, here we can see this is the McConkey's media, and the microorganisms are strict on this media, and we can find the discrete colony or we can find the isolated colonies or the gram negative bacteria which are grown on that McConkey's media only, where this is the TCBS, and this bacteria shows yellow colony. Already we have seen on TCBS media the Vibrio cholerae shows the large yellow colonies. And this is the potassium telluride media which shows the growth of microorganism, and Lowenson-Johnson media, the growth of Mycobacterium tuberculosis. Indicator media, these are the media which contains some indicator ingredients. This ingredient changes its color when certain type of bacteria is present in it. That means, if that bacteria is present, the color of media changes, which confirms the presence of that particular microorganism. This is the example. The Streptococcus pyrogens forms this type of colonies. Likewise, the urease media changes its color from yellow to blue. Then next type of media is differential media. So, these are the media which enables or which can distinguish between different types of media, or we can differentiate different bacteria in this media. For example, McConkey's media. So, you might have wondered that McConkey's media, it is selective as well as differential also. So, why the McConkey's media is selective as well as differential, that we are going to study in next slide. It can differentiate between lactose fermenter and lactose non-fermenter bacteria. So, here as already discussed, the McConkey's media, it is selective for gram negative, means it allows the growth of gram negative bacteria only, whereas it is differential for lactose fermenting and lactose non-fermenting bacteria. Here we can see the bacteria which can ferment lactose, they appear pink in color, whereas non-lactose fermenting bacteria appears colorless. So, those bacteria which are gram negative will grow in this McConkey's media, but in the gram negative bacteria McConkey's media has ability to differentiate between lactose fermenting and non-lactose fermenting. This is how McConkey's media is selective as well as differential. Then sugar media, media containing any fermentable substances, for example, glucose, arabinose, lactose, starch, etc. This type of media consists of 1% of sugar in peptone water. So, in this type of media we can place a Durham's tube or a sodium fusion tube in inverted position in order to detect the presence of gas bubble inside the tube. So, microorganisms which has ability to produce gas can be determined by using this Durham's tube. Selective as well as differential media, McConkey's media already discussed. It is selective to gram negative enteric bacteria because it contains bile salt which inhibits the growth of gram positive bacteria. Because of presence of bile salt in McConkey's, it will not allow the growth of gram positive. So, it is selective for gram negative only. It contains lactose which differentiate between gram negative lactose fermenter and gram negative non fermenter. For example, E. coli which has ability to ferment lactose, so it will show pink colony, and gram negative which do not have ability to ferment lactose, for example, salmonella will appears colorless colony. This is a typical useful testing of water sample for coliform bacteria. Then EMB agar media, that is eosine methylene blue agar media, which contain di-eosine and methylene blue. Okay. So, they inhibit the growth of gram positive microorganisms and it is selective for gram negative, as McConkey's they are also selective for gram negative bacteria, and this media lactose fermenting microorganisms such as E. coli produce black precipitate on EMB agar and their colonies will be either black or possesses dark center. And non-lactose fermenting bacteria such as Proteus or Salmonella or Shigella appears pink or colorless colony. Then mannitol salt agar media, it is media selective for Staphylococcus and differential with respect to mannitol fermenting and non-fermenting. So, in this case the growth of most bacteria other than Staphylococci which are halophilic is inhibited by high concentration of salt in media. As already discussed, the bacteria which can grow in high salt concentration are called as halophiles. So, only these bacteria like Staphylococci will grow in this media and other bacteria will not be grown in this media. The fermentation of mannitol can be seen only in pathogenic species of Staphylococci which can be detected by production of acid leading phenol red in the media to change to neutral red to orange or bright yellow color. Means the color of media or the phenol red changes from red to yellow. If the bacteria is pathogenic, the Staphylococcus which is pathogenic species has ability to ferment mannitol and it changes the phenol red color to bright yellow color. Whereas non-pathogenic Staphylococci produce small colony surrounded by red or pink color due to production of basic byproducts due to metabolism. Then these are the phenyl ethyl alcohol agar which is selective media which inhibit the growth of gram negative bacteria and they are selective for gram positive. The phenyl ethyl alcohol acts to disrupt the lipid structure in the gram negative microorganisms and inhibit the protein synthesis. The growth of gram negative microorganisms on this media tends to be stunned or completely halted. Then blood agar media, it is nutritive media with differential properties in with respect to hemolysis. Then the transport media which is used to transport the media from transport the microorganism from one place to another place. Okay. So, we can use a swab sample here like this and we can transport the media or we can transport the specimen to another place. Anaerobic media, these are the media used to grow anaerobic microorganisms. For example, Robertson cooked meat medium or thioglycolate medium.